William Shih holds joint appointments as Associate Professor in the Department of Biological Chemistry and Molecular Pharmacology at Harvard Medical School and in the Department of Cancer Biology at the Dana-Farber Cancer Institute. He is also a core faculty member at the Wyss Institute for Biologically Inspired Engineering, also at Harvard University. He earned an AB in Biochemical Sciences from Harvard University and a PhD in Biochemistry from Stanford University. After postdoctoral work at the Scripps Research Institute in San Diego, he joined the Harvard faculty. Among other awards, Professor Shih has received a New Innovator Award from the National Institutes of Health. And with that, please join me in welcoming Professor Shih to deliver his lecture on DNA nanostructures as building blocks for research and future medical therapeutics. Professor Shih.
Thanks to Larry for inviting me. I've had a good time so far at dinner with the Philosophical Society of Washington, learning interesting things like the relationship between protein staining and belly dancing, thanks to Dr. Carl Merrill. And it's a great privilege to be able to share with you some of the recent advances from my laboratory and the laboratory of my peers in the field of structural DNA nanotechnology. Now, we're all familiar with the role of DNA as a store of hereditary information, but tonight I'm not going to be speaking about that. Instead, I'm going to be telling you about harnessing the remarkable self-assembly properties of DNA to use it as a construction material such that we can build nanoscale devices. So just to bring everybody up to speed in terms of what does nanoscale mean? Let's imagine, by way of metaphor, that a DNA double helix has a diameter of about 2 nanometers, but just to put that on real scale, imagine we're magnifying it to the point where now the DNA has an effective diameter similar to the width of a human hair, about 1/10 of a millimeter. And what we're trying to do, let's say, is to weave these threads of DNA into structures that are the equivalent of the size of about a thimble. We're trying to devise molecular delivery devices that we can now get therapeutics into a cancer cell. And let's say that cancer cell is about the diameter of about 1 yard. And that cancer cell might be part of a tumor that has many, many cells that is the size of, let's say, a football stadium. And that tumor might be in a patient that if we were to take that adult human being, lie them down on the ground, their body would extend from here to Philadelphia. So that's about the length scales that we're talking about, comparing nanoscale, just a piece of DNA or a small part of a protein, to the size of a human body. So please just keep that in mind. And one thing that I've been amazed by over the past couple decades is the amazing power of DNA in order to achieve self-assembly. And one of the things I hope to convey to you is how amazing it is that we've been able to now start to match the versatility of natural systems in order to build these machines. And why is it that DNA in particular is especially good at this? So for the first 2/3 of my presentation, I'll give you some background information and then tell you about recent developments in the field in building using DNA as the methodology. And then for the last third of my talk, I'll briefly go over a few examples in molecular biophysics and then also potential for therapeutics of this kind of technology. So we can look to natural systems for inspiration. They can do all kinds of amazing things that currently human technology cannot match. And they can do this on a variety of different length scales, down to the molecular. And I think one of the key ideas that's special about living systems is that it's using this very simple paradigm of molecular manufacturing, which is that it generates these linear polymers of amino acids that then will fold up on themselves into some complex three-dimensional shape. And it's that three-dimensional shape that imbues the function on it. It can now specifically recognize other molecules, it can undergo shape changes, it can catalyze chemical reactions. But it's all based on a very simple linear manufacturing model. And it's all through self-assembly. So you have molecules that are floating around, and then they'll bind to each other and carry out their function. So just to give you a couple of examples, this is a rendition of a ribosome. So it's about 25 nanometers in diameter. And this is the machine that in the cells that's responsible for manufacturing proteins. And it's quite a remarkable device because what it does is it takes a linear instruction tape that's encoded as a single-stranded RNA molecule, and then it translates the information from that single-stranded mRNA, or messenger RNA, into a linear sequence of amino acids that builds the proteins. So it's like you have an instruction code for proteins, and here's the factory for building it. On the example on the right, this is you can see a 100 nanometer scale bar. It's a scanning electron micrograph of a bacteriophage. It's a virus that infects bacteria. And what happens is it looks kind of like a nanoscale lunar lander, and it docks onto the surface of the host bacterium with these spindle fibers. And then upon recognition, then what it's doing is it suddenly pumps its cargo that's held in this capsid. It's a DNA genome, and it pumps it against the osmotic pressure gradient into the bacterium cell. And that's a key step towards its replication. So it's like a nanoscale hypodermic syringe. Its purpose is for infection of the bacteria, but hopefully you guys can imagine that this kind of mechanism could be used for therapeutic purposes. Maybe we need something to get drugs into a desired target cell. Here's an example of a nanoscale molecular motor that's involved actually in ATP synthesis within cells. That's the energy source of cells. But what we can do in vitro in the test tube is run the machine backwards. So if we give it lots of ATP, it'll actually burn the fuel instead of synthesize it. But the problem is that it's too small to actually see using something like light microscopy by itself. It's too small. But these authors in Japan had this clever idea, which was basically to glue an extremely long rod to this object on the top, such that when this very long rod that might be something like 10 micrometers long, so about 1,000 times longer, now it's long enough that we could actually see it using fluorescence microscopy. So that's what we're seeing here. They glued this very long rod, and now they can actually watch it spin around using fluorescence light microscopy. And shown below is a molecular model of how we think that this object operates, that you have a bunch of smaller elements that are going to go in this pinching motion, but it's the coordinated pinching motions of 6 of these around in a circle that actually leads to the more complex rotary motion of the device. So the point of showing you these movies is not to remember any of the details, but just to drive home the point that molecular machines are extremely intricate and they carry out very sophisticated functions, but at the end of the day, they're still based, from a manufacturing point of view, on these linear polymers that are then coded to self-assemble into these complex functionalities. So here's a movie that was recorded in the 1950s by David Rogers at Vanderbilt. We have here a neutrophil. It's one of the warriors of our immune system whose function is to go around and neutralizing pathogenic bacteria. These are little red blood cells that it's pushing around. And we can see it's doing all kinds of amazing things. It somehow knows how to track the bacterium. It can change its shape. It can engulf the bacterium and destroy it, which could be very important for therapeutic type purposes. And again, the point is that biology can produce incredible complexity based on this fundamental idea of biopolymer self-assembly. And just one other note, that today's pharmaceutical industry is based on synthesis of drugs that are relatively small and uncomplicated. Typically, scientists like to make things that are only 500 times the mass of hydrogen or smaller. But what I think we can expect to see by the end of the decade, and perhaps even sooner, is that we can synthesize pharmaceuticals, therapeutics, that are more like a complex cell than like a dumb little drug. And that it's only with the full machinery of a cell that can sense, compute, cooperate with other cells, expand, divide, squeeze through tight spaces, that we can really achieve truly revolutionary advances in medicine. Well, that's all well and fine for the end of the decade, at the end of the century, but what about now? Or even what about a couple of decades ago? So I can remember being an undergraduate at Harvard, working on some of this biomolecular machinery, marveling at its complexity, and just dreaming that one day myself and others would be able to know enough about chemistry to recapitulate some of these complex functionalities by ourselves. But at the time, it seemed like an incredible pipe dream. One really didn't have a sense of how long it would take before we'd be able to master this kind of complexity. And what I hope to convey to you today is that we're actually much closer than most of us would have dreamed of 20 years ago. So 20 years ago, the main idea is that, okay, well, we can't build these complex machines. The best we can do is to just try to dissect them. But I hope to persuade you that we're moving along quite rapidly. to actually become doers rather than just teachers. Of course, understanding how nature does things is very important still, but it's just that we should also begin to build our own devices. And so you might have heard of this buzzword, synthetic biology, and we can talk about a synthetic biology of molecules, where we could decide to summarize the goals of the field as follows. So one is, could we figure out how to build with the digital building blocks of nature, so atoms and molecules, to make precise objects of any kind of shape that we want, up to the 100 nanometer scale and beyond. So again, this is about the size of that thimble that I had mentioned previously. Could we use that capability now to construct devices that rival the complexity of the things that we see in nature? And then finally, can we then exploit those devices to do things that are useful for human society? Now, I don't want to give you the impression that we've completely mastered the toolbox. Nature is still much better at us than most of these things. But simply, I want to convey the idea that we're getting closer. And in particular, my field likes to work with DNA. And so the question that I'd like you to ponder is, why DNA instead of something like proteins? So DNA is a complex macromolecule. We're all familiar with this iconic double helix. And we know that we can abstract it as this ladder-like structure if we unwind it, where we have one strand that goes in one direction, and then we have another strand that goes in the opposite direction. So that's the anti-parallel nature of DNA. And it's very complicated, but the good news, and why we really care about this for nanoconstruction, is that you don't need an advanced degree in chemistry in order to build with DNA. And the reason why is that it has a very simple and robust rule in terms of how it assembles with other strands of DNA. So one is, I just mentioned, there are these anti-parallel strands. The second one, which is probably the most important one, is that DNA comes with 4 different letters of the alphabet, A, C, G, and T, and it has this very robust code which says that A really likes to pair with T and doesn't like to pair with the other 2 bases, or other 3 bases, really. And C really likes to pair with G and none of the other 3 bases. And so on and so forth for the 4 desired base pairs, which means that if you make a strand of DNA, any random sequence, and then now you present it with a perfectly complementary sequence that has exactly the right sequence obeying this rule, then it will want to pair with that other strand to the exclusion of anything else. And no matter what, it's always going to form into a double helix that's about the same structure, that has a reproducible twist of on the order of about 10 units per turn. So in other words, we have this very simple rule for how these strands come together, and we know very reliably the structure that they come together to form. And this is in contrast with something like proteins, where there does not exist such a simple code. The closest you could imagine coming to is something like antibody-antigen interactions. And in that case, it's not something you could just write down on a piece of paper. And furthermore, the geometry of how they come together is different for every antibody-antigen complex. So it's really the simplicity and robustness of DNA self-assembly that gives it its advantage for nanoconstruction. Because if you have something that's simpler, that means for the same amount of human effort, you can now generate objects of much greater sophistication. So here's the founder of the field, Ned Seaman. He invented structural DNA nanotechnology in the early 1980s. And his dream was to use this as a tool for something called protein crystallography. Because it's been known for a long time that you could decipher the nanoscale structure of proteins by this trick, where if you could coax the protein molecules to form a regular crystalline array, then you can now tease out the structure by illuminating that crystal with X-rays. Otherwise, it's very difficult or close to impossible to see these things that are too small to see using a conventional light microscope. And he was frustrated by the slow pace of this procedure, and eventually he had a eureka moment while he was sitting in the campus bar, which was that this woodcut of M.C. Escher popped into his head, and suddenly he thought, oh, I got it. Now I can replace these flying fish with DNA branch junctions, because that's something he had been working on. So it turns out you could basically program DNA to make these branch junctions, and he had the idea that he could link them together to form this rationally designed porous crystal, and then he could dock those proteins that he wants to solve into stereotyped locations within each unit cell, and then basically use that rational crystal to force crystalline order on the guest protein and make the problem easier. Well, he's been working on this for a number of decades. This was his first big experimental breakthrough about 10 years later, in 1991, he reported a DNA cube, wireframe cube, where every edge is 2 turns of a double helix, then every face is a circular strand of DNA. This is about 10 nanometers in diameter. And what I'll be showing you is that in the intervening 20 years, we've made an incredible amount of progress in building objects of much greater complexity. And it turns out historically, what's been the most important for catalyzing this advance in complexity of structures is this idea that Ned stumbled upon, which is that he could effectively glue together adjacent double helices with these crossover motifs. So if you were to just build with linear DNA, then that's kind of boring. You can make it longer, you can make it shorter. The excitement from a structural point of view is when you can make branched structures, and especially rigid ones. And so what he found is that if you programmed a strand to be part of one double helix, but then to jump over to another double helix, then he could, and if he did that with gluing it together at multiple spots, then you could create these rigid Lego-like bricks that would now allow you to make an advance on 2-dimensional and 3-dimensional structure. And another idea about this that's exciting is that it's just using regular plain vanilla DNA. So what we're doing is, from chemistry, we've learned, after a Nobel Prize, we've learned how to synthesize DNA strands of any sequence we want. And then we could just basically program one strand to be this sequence coming across. It could be some random sequence. And then likewise on the top. And then we could synthesize another strand that's programmed to be the perfect Watson-Crick complement. Again, A pairs with T, C pairs with G, such that it's following a path in the bottom helix. And then, like a molecular square dance, it switches partners, and then now pairs with another strand. And then that way, we very reliably can force the DNA to make these intricate structures. Once you have this primitive building block, then in principle, you could imbue the ends of it with single-stranded overhangs, known to molecular biologists as sticky ends. And you could program many of these in a test tube to link up in very specific fashion. So let's say we have, in this example, there's 2 kinds of Lego bricks. And we make sure that this sticky end on the upper right-hand corner of the red brick is only complementary to the sticky end on the lower left-hand corner of this purple brick, and so on and so forth. So in this toy example, if we added these 2 together, then you should ideally form this 2-dimensional lattice according to those rules. And that actually works in a test tube, which is quite remarkable. The idea doesn't quite extend in terms of making more complex objects. So what you could ask the question, well, what if instead of just 2 kinds of bricks, what if I had 1,000 kinds of bricks? Would they actually find each other, find the correct partners? And it turns out the answer is no. But what I'm going to be telling you is that there are similar methods where the answer is yes. And I'm going to go through some of the details of that next. So the first method that was really successful in taking this idea of gluing double helices together in a parallel fashion, but then really advancing to complex structures, is known as DNA origami. It was developed by Paul Rudiman at Caltech in 2006, and the idea is as follows. So you start from the 7,000 base genome of the M13 bacteriophage. It's, again, a virus that infects bacteria. It has this genomic single-stranded DNA of known sequence. And as far as we're concerned, it's kind of a randomly generated sequence. We like it because we can get our hands on it. And based on that known sequence, we now chemically synthesize these programmed so-called staple strands that are 20 to 60 bases long, that through this Watson-Crick interaction, A pairs with T, C pairs with G, is forcing that long strand into this anti-parallel set of loops. After mixing all these 200 strands together, heating up to 60 degrees centigrade or higher, and then cooling down to room temperature over the course of about an hour. And at the end of the process, what we've done is we've programmed the system to form a structure that looks like a parallel array of DNA double helices, kind of like a molecular log raft, or a molecular version of one of those maki sushi mats that you use to roll up your maki sushi. Each double helix has 1 strand contributed by this gray scaffold strand, the long strand, and 1 strand contributed by these colored staple strands. So we might have one that starts on one of its ends, the so-called 5' end, and then it's moving in this example from right to left, base pairing. And then we program it to switch through a single phosphate linkage now to the adjacent double helix, and now it's going from left to right. And again, this is working because that gray scaffold strand has unique sequences along its entire length. And therefore, the staple strand has a unique partner to link two segments together. Paul Rudiman used this method to build a diversity of complex 2-dimensional structures. This is an atomic force micrograph. It allows you to see things smaller than the wavelength of light. And the dimensions of this object are about 100 nanometers by 100 nanometers by 2 nanometers. They called it the disk with 3 holes. And this example in the upper left-hand corner represents in size just part of the upper lip. And by the standards of most molecular scientists, including molecular biologists, this is a massive structure. And most of us were astonished that something like this could be programmed. We could program something of this complexity to form using such a simple idea. It's about 5 megadaltons, which means it's like 2 of those ribosomes that I showed you before that you could program to mash into any kind of cookie-cutter 2-dimensional shape. But because it's self-assembly, we're actually making trillions and trillions of copies of these devices simultaneously. So we're adding in trillions and trillions of copies of the building blocks, and they all find each other and make this thing. And in fact, Eric Winfrey, Paul's advisor, made the joke that at the time they had created the most concentrated form of happiness known to humanity. They had trillions of copies of this in just a droplet of water. So when I first saw this, I was filled with equal parts amazement and envy, and I wanted to contribute to the effort by extending to 3 dimensions, but first I'd like to just go through some of the examples of what Paul was able to build. So what he's doing is he's taking that same M13 genome, and in different test tubes he's adding different sets of 200 strands to fold it into different structures. This is a simple rectangle, and then the color coding represents basically the raster path of the strand through the object. You can build a little star shape, so it's basically weaving back and forth like this, going through the structure. You can actually write things onto these structures. So the idea is that if you make one rectangle where you have no surface features sticking out, so it's kind of a flat surface, and then you make an identical set of strands where you're putting some kind of surface feature on top of the device, then you can think of that as a thick brush on top. And then now the fun thing that he can do is to mix and match strands, because every strand has a unique 2-dimensional location in this tapestry. So at each pixel, he can decide whether he wants to have a strand that has no feature versus a strand that does have a feature. It's very analogous to a pixel in computer graphics. So now just by mixing and matching strands, he can create any pattern that he wants. So here he made DNA. These things turned out to be sticky, so they polymerized into this long double helix. He made a map of the Americas, and then Paul's very modest, so he apologized profusely for being so America-centric and not having the time or resources to make Europe or Asia. You can also connect things at slightly different angles to some limited success. This method works around the world, not just at Caltech. So colleagues in Aarhus, Denmark, decided to implement their school emblem, these 2 dolphins doing maybe some immoral things to each other, I'm not sure what, but they showed this proudly to the provost of the university, and the provost said, "Oh, you didn't hear, we changed the emblem of the university." So it's no longer the emblem of the university. Lulu Chen was a student in Shanghai, and without contacting them, she just read the paper and then designed a map of what she thought was supposed to look like China, Taiwan on the world's shortest leash, and was able to build this structure. She used that as basically her application to a postdoctoral fellowship at Caltech, which she got, and now she's been promoted to assistant professor at Caltech. Okay, so what about, so one of the criticisms of DNA origami is, well, you're taking a strand and you're making a sheet. That doesn't really sound like origami. Origami is taking a 2D sheet and folding it into 3D structure. And I think it's kind of a semantic issue, but we should go ahead and make those people happy. So we can take inspiration from macroscale origami, where if you really take it to an extreme, you can make very exotic structures that require some computer algorithms in order to help you out. Believe it or not, it's just one sheet of paper can make one of these objects. And that's amazing, and what we hope to do over the next couple decades is match this level of sophistication using DNA origami. We're not quite there yet. So what can we do with DNA origami? So that same group in Aarhus, Denmark, they're able to fold that M13 genome into a series of 6 small sheets that they then program to fold up into a nanoscale box that's about 60 nanometers per dimension. And they even had a little lid that they could program to open and close upon input of single-stranded DNA input. And the idea was to create a kind of toy example of a drug delivery box that you could imagine has some kind of therapeutic cargo on the inside, and only when it gets to the proper damaged tissue, it'll recognize some molecular signature that will cause the lid to pop open, and then now those therapeutics will specifically come out, and they won't damage healthy tissue. That's the basic idea. All right, so in my lab, we wanted to extend to 3D by building solid three-dimensional structures, because we believe that these single sheets of DNA, they're very interesting, but for some applications, they're too floppy. Just like if you wanted to build a house, you might soon become dissatisfied with the idea of building the house out of sheets of paper, because it won't be very stable. You might be more attracted to the idea of building your walls out of reinforced cardboard, because it's much thicker and more rigid. And maybe that's still not quite rigid enough for you, but it's better than paper. So how can we make thicker structures out of DNA? And the idea is as follows. So imagine that, again, here we have a model where we have these DNA double helices, and they're linked together by these crossovers, these strands crossing over from one helix to the next. And I represent this as a flat sheet, but it turns out by shifting the position of these crossovers, we can program the sheet to curl up. So for example, if we move these crossovers on the top 2 base pairs to the right, then hopefully you can imagine that the sheet is actually going to curl, actually those 2 bases to the right, to the left. We can imagine that this sheet is actually going to curl towards the back of the plane of the page, and that has to do with the helicity of the DNA. And likewise, if we shift those crossovers 2 base pairs to the right, then that's now going to force the curvature towards the front of the plane of the page. And the take-home message is that simply by changing some of these subtle design features, we have absolute control over the curvature of these sheets. So here's another example. Each one of these cylinders represents one double helix of DNA, so it's like the example in the upper left-hand corner, but rotated by 90 degrees. And what we're doing is we're shifting around the position of those crossovers so that the sheet no longer prefers to be flat, but instead prefers to curl up. And in this case, in this example, we're programming it to curl up into this corrugated S shape. And furthermore, anytime you have helices that are touching each other, we're programming staple strands to be going through the interface between those helices. So we might have one staple strand go 7 base pairs here, crossover 7 base pairs here, 7 base pairs, 7, 7, 7. So these staple strands are really gluing all of these helices together such that if this process works, then we have a highly cross-linked object. So we knew from Paul's experiments that you can make these 2D structures, and we wondered whether or not physics allows us to just stack them up to make solid structures. And with those of you who have a background in experimental science, you probably can predict that the experiment didn't work, that something prevented it from working. And you could imagine that, well, maybe you can't just force all of this negative charge into this small three-dimensional space. But we were persistent, and eventually Hendrik Dietz in the lab figured out that it wasn't that it's not thermodynamically stable, but it's simply kinetically difficult to achieve. And so we found that eventually that we could get success instead of heating and cooling over the course of an hour. Instead, if we heated and cooled the object over the course of a week, then we could finally get reasonable yields of the object up to about 50%. And so Sean Douglas in the group built, for example, an object instead of with 3 layers, he built something with 10 layers. And then he added all the strands together, heated it up, cooled it down, and then looked at it using the transmission electron microscope, where it's another method where we can see molecules. And we see something that's about the right dimensions, 14 nanometers by 36 nanometers by 36 nanometers. And so now I'm just going to go through a gallery of different objects that we built. These are all 20 nanometer scale bars. Again, that's about 10 times the width of an individual DNA double helix. This one we call the monolith. It's like a nanoscale crystal, but it's important to remember that every single element of the object has a unique sequence associated with it and therefore is independently addressable. We can put functionalities anywhere on this object that we want, which is a little bit unprecedented so far in the world of synthetic nanoparticles. Here's the object built by Franziska Graf when she was in the group, a graduate student. We called it the genie bottle. And one version of it, we actually didn't fold part of that long scaffold strand. So in the microscope, it looked like little wisps of smoke coming out of this nanoscale genie bottle. Here's another object built by Sean. It has a 7 nanometer hole in the middle, so it's like a nanotube made of DNA. And we could program sticky ends on one face to be compatible with sticky ends on the opposite face. And then when we mix them together, it'll form this very long polymer, homopolymer, that's structurally reminiscent of some of the thick filaments inside of cells, such as actin filaments or microtubules. Here's the object built by Tim Liedl when he was in the group. Again, these helices, these cylinders are supposed to represent DNA double helices, and you can imagine if we take cross-sections through the object, then it's a different arrangement of double helices through each cross-section, which I think reminds us that from a design perspective, it's kind of like sculpture in the sense that you have a solid block of material, and then you're chipping away at that solid block until you get whatever structure you want in the end. But in this case, we're actually compiling that 3D structure into a series of these strands that again, self-assemble into the final three-dimensional structure, which has some advantages, as I mentioned previously, like we can scale this. We can create trillions and trillions of these molecules simultaneously. Here's the object built by Bjorn Högberg when he was in the lab. We call it the slotted cross. There's a H-shaped domain and an O-shaped domain, and the middle of the O of the H domain is threading through the middle of the O domain. So we get this very complex, almost ship-in-a-bottle type assemblies, and it still works just through the magic of self-assembly. Here's another kind of crossed object built by Hendrik Dietz. So we can take a little bit of a closer look at this slotted cross. So this is a stylized rendition of the passing of the scaffold strand through the object. You can see it looks very complicated, but it actually follows from a simple principle that I'll share shortly. We can see the H domain is linked to the O domain through these little connections here. And this is the kind of diagram that we work with on a day-to-day basis in my laboratory. So we call it the strand diagram representation. And it's basically a two-dimensional representation of the object, where we have the scaffold strand is in blue, it's going back and forth, and we can see the staple strands linking things together. But then actually in 3D, that two-dimensional map is blueprint is kind of rolled up into 3D structures. So for example, we might have a staple strand that starts on helix 8, jumping over to helix 7 to 6, and then it looks like it's making a big jump from helix 6 to 1, but in fact in 3 dimensions, those helices are designed to be close together, so it's actually not a big jump. And the point I'm trying to make here is that although it's a complex strand wiring, when you squint at it, it looks like a blueprint. So you could imagine that this actually is an H-shaped domain, and this actually is an O-shaped domain. So we have this correspondence, sort of correspondence between our representation of the object and the actual three-dimensional structure that we're building. So again, just to review, we can use DNA Origami to make these sheet-like structures, but in fact, we're not forced to make them planar. We could program them to curl up into some kind of accordion-type shape. What I just showed you is folding them into this accordion, to this honeycomb lattice-type shape, kind of crinkled shape, but it turns out that we can also fold them into something that some of you might find more comforting, fitting a Cartesian lattice. But both of these basically work. I think it's important for any kind of molecular manufacturing to have software tools that can help reduce the barriers to implementation. So Sean Douglas had a computer science background, so he spent a year developing a program with a graphical user interface to build whatever structure that you want. So here we're looking down the axes of the double helices, so you can select which strands that you want to include or exclude from your overall block of sculpture. If anybody's interested, you could download the software for free at cadnano.org. So here's the strand diagram representation that we spend most of our time on, and then there's also a little window that gives you a three-dimensional representation of the object. So what about building larger structures? Well, one idea is to just add more unique strands, which we're interested in, but we'd like to combine that with other well-known structural principles. So one is that you can build high-strength weight objects by constructing wireframes. So here what we've done is we've programmed that long scaffold strand to fold up into a branched tree structure where each one of these struts is 6 helices, so it's about 7 nanometers in diameter. And then we program sticky ends on these terminal branches to link up in a defined fashion to make these double triangles. And in 3 separate test tubes, we're folding that M13 into 3 different double triangles that superficially look the same, but they have different arrangements of sequences. So they all have unique sticky ends. And we program those unique sticky ends to link together in a very unique fashion, which is to make a wireframe icosahedron. So here's a macro scale reenactment of the self-assembly process. This is guided self-assembly on the macro scale. But the good news is that it works even without hands, and it works on a much smaller length scale. So here we're looking again using transmission electron microscopy. The objects are predicted to be about 100 nanometers in diameter. And this is a 1 micrometer scale bar, so that looks like they're about the right size. You can see there's some misassemblies as well, but we're glass half full kind of folks, so we're glad that anything forms correctly at all. Zoom in, 500 nanometer scale bar. We can see the kind of characteristic hexagonal outline that represents the projection of icosahedron. Zoom in a bit more, 200 nanometer scale bar, so we can make out these triangular type faces. There's certainly something lattice-like going on. There's also some misassemblies as well. And then here, this is the highest effective magnification we can go to, 100 nanometer scale bar. We can make out these triangular faces. We can make out, or think we can make out these five-fold vertices. So it really looks like we're successful, at least for some of the particles, in building a wireframe icosahedron that's the same size as a medium-sized virus, which, again, that's something that 10 years ago would have seemed ludicrous. But in fact, we can do that just by following the simple rule of A pairs with T, C pairs with G, and then taking it to its extreme. So it's a little bit analogous to when we look at computer programming, we just have zeros and ones. But if you have enough of them, they can do amazing things. So in this orientation, this object, you expect to see X, 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 which is what you see. We're inspired by art as well as practical things. So there's this guy, Ken Snelson, who developed these floating compression structures. He was inspired. I saw a photograph of one of your alumni, Buckminster Fuller. So Buckminster Fuller was lecturing about tension and compression, and then Ken Snelson took these ideas and made these interesting sculptures. We have these beams that aren't touching each other directly, but instead they're connected by these cables that are bearing tension. And if you arrange them in the right way, you create this extremely high strength to weight object that has some interesting mechanical properties, such as when you apply a global load to the object, then those rods will rearrange in three-dimensional space, and then when you relieve that load, then it'll pop back to its original shape. So Ken Snelson only thought of this as something interesting from an artistic point of view, but other scientists have looked at this and thought that this is actually an elegant way to transmute between forces in chemistry on the molecular scale. And my colleague Don Ingber has done a lot of work in basically demonstrating that cells, in fact, do use this principle. And as a first step to making some synthetic materials that we could use to interface with cells in interesting mechanical ways, for example, that might be useful for tissue engineering and regenerative medicine, we wanted to implement this kind of strategy with DNA. And what we're able to do with working with Tim Liedl was to add staple strands that would fold that long M13 scaffold and then fold it up into a structure where you have these three rods, each, in this case, there are 13 helices, and they're linked up with single-stranded DNA that's acting as a tension-bearing cable, and therefore folding these kinds of structures. And here are some transmission electron micrographs of the objects. These are simple two-strut kites, and then some of these other objects have the three struts. So in this way, we were able to succeed to build these tensegrity structures, reduce it to the molecular scale, and we think that this could be useful for tissue engineering type applications. So that was kind of a combination of tension and compression. Hendrik Dietz was also able to implement this, but within a more primitive structure, just this bundle of DNA double helices. Again, every cylinder is a double helix that we're cross-linking together. And so what he wanted to do was to make bent structures. And the idea was as follows. So if you reason that these DNA helices are kind of these elastic type structures, then he said, well, what would happen if I made the helices a bit longer on the top? So I made extra units on the top, and then I removed some units on the bottom, so individual base pairs. So what would happen is the top would be under compression because you're putting more material in the same amount of space, and then the bottom would be under tension because you have less material occupying the same amount of space. And the mechanics tells us this system is going to relax if it's an elastic material by bending. Actually, I had it backwards, so there's shorter helices on the top and then longer helices on the bottom. And so he implemented this with an 18 helix architecture. So the curved region is in red, and then he has these straight regions on the side just so he can visualize it. Again, these are 20 nanometer scale bars. So here he has a relatively modest amount of shorter helices on the top and longer ones on the bottom. You can get a predictable 30-degree bend. You have twice the amount of perturbations. You can get a 60-degree bend, 90-degree bend, 120-degree bend, 150-degree bend, and then all the way to 180 degrees. So this is basically straining the DNA quite a bit, and it's almost as tight as DNA wrapping around the histone proteins within nucleosomes. That's the way that our cells basically wrap DNA into tight little bundles. And that's done by proteins, but in this case, it's being done just by DNA base pairing. So I'm just going to skip through this slide here, show you some other examples. So they had some fun here. Here's 6 semicircles of increasing radii of curvature. Here's a little curved beach ball. Here's some recycling, some green technology. So he took that 120-degree boomerang from before and programmed sticky ends to link the front and the back, and then it'll homotrimerize because it's rigid. This has become a symbol for synthetic biology, meant to just be an abstract notion of technology, but Hendrik decided to implement this in molecules. So he programmed a beam to bend into a 180-degree angle with 3 square-shaped teeth, and then at the front, sticky ends compatible to the back, then it'll decide to make a rigid homodimer that's 50 nanometers in diameter. If you make one that only bends by 90 degrees, then it prefers to make a tetramer. So we thought this was maybe the first molecular example of gears, but then just opened up a Science News article, actually online, a couple months ago. So a group found that there's these insects that have these gear-like structures in their legs that help them to jump up. So that just goes to show that nature usually figures things out a few million years before we do. Okay, so all this is basically using some long piece of DNA that's being folded into a complex shape. And we have toys that remind us of this principle. They're quite versatile, we can make all kinds of shapes. But I think most of us were imprinted at a young age to use a different kind of principle that seemed more versatile, which is to have little modular building blocks, or at least the Lego sets of my childhood were mainly these kind of stereotype bricks. The ones today, I think each one's a different shape. But back when I was a kid, then they're all the same shape. And you can now link them together in myriad ways to create any kind of structure, and you're not constrained by this long thing going through the entire shape. So it just seems more aesthetically pleasant. But as DNA nanotechnologists, we all came to believe that that long scaffold strand going through the structure was very, very important, because nobody had been able to make something of that complexity without using a long strand before. But my colleague Peng Yin at Harvard decided he wasn't going to have any of this dogma, and along with his students, Brian Wei and Mingjie Dai, demonstrated that you can make a structure the same complexity as DNA origami, but without that long strand. So the basic idea is that each one of those standardized building blocks has 4 different domains, and its structure within the final tapestry is such that the first 2 domains are part of one double helix, and then the second 2 domains are part of an adjacent double helix. And these tiles or bricks know where to pack within the lattice based on that Watson-Crick base pairing. So you design the sequences such that each one of these bricks has a unique sequence and has a unique set of 4 nearest neighbors. So for example, brick U4.2 is only going to be compatible with U5.1 along this axis, so on and so forth. So every one of these bricks has a unique sequence and therefore has a unique location within the lattice. And the amazing thing is that this actually works almost as well as DNA origami. And so you can make this 2-dimensional tapestry, and one of the cool things is now you can make a new structure. You don't have to reorder the strands, you're just repipetting them. So anywhere you want a hole in your tapestry, you just leave out that strand. You still add it to the self-assembly mix. So in that way, students were able to build these 100 different shapes just by leaving out strands anywhere they want an eye socket or mouth or curvature. So Yonggang Ke is a student in my lab, and he decided he wanted to pursue extending this DNA tile or brick idea from 2D to 3D, just as we had done previously with DNA origami. So he collaborated with Luvina Ong and Peng Yin to implement 3-dimensional structures with this brick idea. And so this is an abstract representation of this tile that I showed you before. Because each one of these domains is one turn of a double helix, that basically determines the geometrical constraint of these things linking together such that they'll form a planar object. In order to get to 3D, we need something that's not a planar interaction. So it turns out all you have to do is to shrink the size of the domain from 1 full turn of the duplex down to 3/4 of a turn. And so now these structures, instead of fitting together in a planar fashion, they'll only interact at this 90-degree angle. And if you have a bunch of Lego bricks that can only interact at a 90-degree angle, hopefully you can imagine how if you had a whole set of these bricks, you can actually tile a three-dimensional space. So it's the same idea as in 2D, but now it's in 3D. Each one of these bricks has a different sequence, has only 4 unique nearest neighbors based on that base pairing complementarity. From a design point of view, what we can do in a computer program is we could abstract this to a series of volume elements or voxels. Each one of these is 8 base pairs, or that's 3/4 of a turn of the duplex. It's the interaction of 2 of these bricks together. So we can imagine in our CAD program, we can chip away at this to make whatever 3D structure we want. Then we can compile that into a series of bricks that we're either adding or not adding to the self-assembly, and then that gets passed on to pipetting instructions to a pipetting robot, or to a graduate student if you want to do things the old-fashioned way. And then you fold it, look at it using electron microscopy. So here's the different designs. Again, it's the same master set of about 1,000 strands, but just repipetting them 100 times in different arrangements to get new structures. So we can see intricate cavities and things being inscribed into these objects, Chinese characters for those of you who can read them. This row is a unique representation where the solid actually represents bricks that were left out of the assembly, and the translucent represents ones that are left in. So in this case, it's supposed to represent completely enclosed molecular cavities. We can see three-dimensional versions of Roman numerals. We can see little spaceships. And then the transmission electron micrographs of the objects. So again, we can see letters, little spaceships. This is an artist's rendition from Gail McGill of what we think the dynamics of something like a spaceship would look like. And the cynical joke for our field is we can see how far we've come, that 7 years ago we could only make smiley faces, and now we can make spaceships. But and at some point it seemed the funding agencies get suspicious, and it looks like we're having a little bit too much fun. I don't want to dwell on this. So there's 2 different methods, DNA origami and DNA bricks, and they can roughly provide complementary capabilities. It could be an interesting discussion topic for afterwards. I can just say that my lab continues to prefer to use DNA origami for discrete objects. And so for the last part of the presentation, I'd like to discuss a few of the applications that put our funding agencies more at ease, that we're not just wasting taxpayer money. And one area is as tools for molecular biophysics. So as basic scientists, we're interested in understanding what exactly is the shape of these biomolecules and how is it that they interact to carry out the fantastic functions of the cell. And one of the outstanding questions has been, what is the precise 3-dimensional configuration of atoms in these machines? And it turns out for technical reasons, it's very difficult to answer this question, and it's doubly so for a particular class of proteins known as membrane proteins that reside basically in the bubble-like shell that covers all of our cells. These membrane proteins oftentimes serve as gateways in and out of the cell, and in fact, the majority of drugs that we currently use are interacting with these membrane gatekeepers. And yet we don't have a good method for figuring out their structure. We'd like to, because if we knew the structure, then we could design drugs that could muck up those machines. It's hard to do if you don't know what the structure is. This is a really important problem for biomedicine. And so again, Ned Seaman, his dream was to use DNA nanotechnology to solve this problem by this so-called host-guest crystallography that I explained previously. It's been a difficult problem, and they have had some success. So a few years ago, they published this rationally designed crystal where each unit cell is these 3 double helices going up and under each other. And they can self-assemble this into a macroscale object that's about 0.2 millimeters per dimension. But it still doesn't diffract quite to atomic resolution. And we still have the problem of getting these proteins into each unit cell in a rigid fashion. So we've made some really nice progress, but we have a long ways to go. And I think it's fantastic that we have this very difficult goal to pursue, because I think that's how progress is made. You have ambitious goals, but it might still take us a while to get this perfect orientation of all the guest molecules. And what I'd like to tell you about tonight is that you can do something technologically much more modest and yet still have the practical benefit of helping you to solve atomic resolution structures of membrane proteins. And this is a phenomenon related to NMR, nuclear magnetic resonance, that's known as weak ordering. And I'm not going to explain to you the details of how this works, so you shouldn't be able to understand this in full unless you already understood to begin with, but I just want to give you a flavor of what we've been able to do to solve this problem. So again, the goal is to solve the structure of membrane proteins. And it turns out, for various technical reasons, what we're trying to do technically is we have our molecule of interest that we're trying to image, and we want it to be randomly diffusing around in solution, but we want to embed it within a matrix of these molecular trees that are aligned, but only about 2% weight to volume. And we want to do that because the protein of interest, because it has some asymmetric shape, it's going to be slightly more likely to collide with these aligned molecular trees with its long axis perpendicular to the long tree rather than parallel. And the net effect of this transient interaction between your non-spherical protein population and these molecular trees is you get a slight orientational bias in the sampling of orientations of those target molecules you're interested in. About 0.1%, it's pointing in 0.1% more in one direction than in the other principal orthogonal axes. And so if you can impose this slight anisotropy on the structures, then you can now record high-resolution data encoded in what are known as dipole-dipole couplings, and then use that to deduce the high-resolution structure of that protein. So in a very indirect way, our nano trees that we're going to build from DNA nanotechnology is, metaphorically, is like a molecular microscope into the structure of these proteins. That could be very important for medicine and biology. So we did that, and we demonstrated that it works, and then the next step was we collaborated with James Chu and Marcelo Berardi at Harvard Medical School to apply this tool to solve the structure of UCP2. It's a protein that was previously of unknown structure that sits on the inner membrane of mitochondria. It's actually a proton pump, and it's important for some energy metabolism type of mechanisms. I don't have time to really go through the details of exactly how it works. I just want to drive home the one point that we have our DNA nanotubes that can serve as these molecular trees, and we use that to solve the structure of a membrane protein. And this is something that was previously unprecedented. So one reason why I like to give this as an example is that it represents a tool that actually works. And most of the tools, I have to give a confession, that we try to propose are more works in progress, that we might not actually see practical success for a few decades. But in this case, this is something that works today and that structural biologists can use today. So the second area in terms of tools for molecular biophysics that I'd like to briefly touch on is something that I like to call breadboard biochemistry. And the backdrop is that there are many phenomena within cells that involve the cooperation of many copies of different protein players. And as biologists, we want to understand what is the mechanism of this phenomena. So for example, we saw examples of something that will penetrate into cells or something that will manufacture proteins. So how is it that these different components can interact with each other to carry out this process? And I think one of the strategies we'd really like to take to solve this problem is to set up a molecular theater where we can control exactly how many actors of these protein actors can participate, and maybe also constrain them in terms of their geometry with respect to each other. And with this added level of control, we think we can tease out the mechanism of activity of these proteins. Those of you who have some background in biochemistry, this might look kind of unfamiliar because traditionally biochemists, they just have their solutions of proteins and they mix them together and everything happens in bulk solution, and they hope they can deduce what's happening. And I think the next generation is really teasing out what's going on at a single molecule level. So just very briefly, this is an example of breadboard biochemistry that we did with my colleague Samrek Peterson at Harvard Medical School. So here we're trying to study the collective action of motors that are acting on the inside of a cell. This is a class of what are known as microtubule-based motors. So cells have this kind of rigid backbone of microtubules crisscrossing around the cell, and it has these motors that basically provide the force for pushing around cargo in and out of the cell, around the cell. And one of the surprising things about this is that you have these motors that some of them will move in one direction along a microtubule. So imagine we have a microtubule substrate down here. And these are like little race cars moving along that microtubule highway. Some of these motors, these dynein motors, will move towards the minus end of the microtubule. And some of the motors, these kinesins, will move towards the plus end of the microtubule. So it seems a little strange that you have these two kinds of motors, they want to move in opposite directions, and yet somehow the vesicle decides which way to go. So in order to start studying this process, we decided to create this artificial model system where instead of an actual vesicle or bag of cargo, we're replacing that with a DNA origami chassis. This one in this example is 200 nanometers in length. We put some fluorescent dyes on it so we can see it using light microscopy. And then what we're doing is we're decorating the object with these single-stranded DNA extensions, and we can put any sequence we want at each location. And in this particular example, we're putting a strand of one sequence at these two locations, and then a strand of a second sequence at these different 5 locations. And then what we're doing in the second test tube is we're preparing this dynein motor with a complementary DNA strand such that it'll hybridize only to these first 2 positions. And then in the third test tube, we prepare this kinesin motor with a complementary DNA covalently attached to drive its self-assembly at only these 5 positions. So we can control the stoichiometry, so the number of motors and the geometry on the structure. And in this initial study, what happens is when we dump this chassis down onto microtubules, you can ask, well, who wins? Does kinesin take over because there's 5 of them? Or does dynein win because it's bigger and stronger? And what we found in this case is, at least for this artificial system, when we put them on the microtubules, they just sat there and didn't do anything. So these guys wanted to move left, these guys wanted to move right, and they just weren't going anywhere. And what we could do experimentally is if we then transiently release the green guys using, we program this photocleavable linker, so we shine a UV laser on this, and then that will release these green guys. And after we release the green guys, then now the orange ones take over, and then this chassis starts marching towards the plus end. And likewise, if in another experiment, if we photo-release these orange guys, then now the green ones can take over, and this assembly will start to march towards the left-hand direction. So we didn't really answer the question of how do cells regulate the direction that these vesicles are moving along the microtubules, but we think we've taken the first step to provide an experimental foundation that we can start to answer this kind of question. So for the last 10 minutes of my presentation, I'd like to briefly go over some potential applications towards therapeutics. So I just want to preface by saying we haven't cured cancer yet, so please don't be disappointed when I reach the end of my talk and cancer is not cured. But we're trying. So one of the ideas is that you want to have your kind of therapeutic payload, maybe it's some kind of chemotherapeutic or radioisotope, and you only want to kill the damaged tissue and you don't want to kill the healthy tissue. So one idea is we could package these kind of therapeutics into little nanoscale delivery devices, and if they could specifically target the diseased tissue, then maybe there'd be much less collateral damage. So along that route, you might want to ask, well, how can I convince these little particles, packages, to go into the right cell and do it efficiently? So one of the things that might affect that is the size of the package. So here's a metaphor of getting things into places where they're not wanted. Katrina Galkina was an intern in the lab. When she came in, she didn't know anything about DNA structure or computer programming, but because we had this CAD-based program, within one afternoon, she was able to design this structure here, added all the strands together, heated it up, cooled it down, and after a week, this is what she saw in the electron microscope. We can see that this one seemed a little bit happier to see her than this other one. That's because she had a very thin linkage between this tail and the body, and just mechanically, that becomes not that stable. Hendrik Dietz, after he left my lab, he started his own lab in Munich, and maybe to celebrate his liberation, it was close to Oktoberfest, he made this dancing robot. And the point here is not that we think that these particular structures will be excessively good at getting into cells, but they serve the function of reminding audience members that we do have this precise control over structure, and maybe there is some specific structure that would help us towards this goal. So what is it about structure that might help? Well, if you think about it, there's a huge universe of possible shapes, and it would take a lot of work to comprehensively answer that question. But just as a first step, as a pilot study, we wanted to look at one simple descriptor of shape, which is aspect ratio. So we made these eight different structures. These blue ones are all 5 megadaltons in size. These orange ones are 2 megadaltons. But let's just focus on these ones highlighted in yellow. Again, they're all the same mass, but by programmable self-assembly, we could form them into different shapes. So elongated ones that are thicker, and ones that are more thick and very short. And we can ask, do these structures get in more easily when they're compact, or do they get into the cells more easily when they're extended? So we're looking at the endothelial cell line as our model cell system in culture. And what we found, at least for this cell line, was that the more compact structures got in much more rapidly. So up to 6 times faster than these extended structures, and up to 20 times faster than plasmid DNA of the same molecular weight. But we also saw interesting behavior from these extended ones, which is that they would tend to stick to the cell surface, but then resist getting internalized. So it's kind of like if there's a cat that's perched over some kind of surface, and then you're trying to get it in, but it won't go because it's made its body so long. And we think this could be useful for medicine because what if you wanted a two-tiered strategy where you have a first wave of nanoparticles whose function is simply to mark the cells for destruction. So let's see, the first wave basically marks the cancer cells for destruction and doesn't mark the healthy cells. And the second wave of nanoparticles uses the cue from the first set of nanoparticles to decide who to deliver the therapeutic payload to. Well, it would be a problem if that first set of nanoparticles just got internalized by the cells, because then they would be invisible to that second wave. So we think maybe that might be a useful strategy for creating sentinel nanoparticles. We can also decorate these objects with lots of ligands. So in this case, we're using this single-stranded extension hybridization method to put these hairy extensions on the object, in this case decorated with a cyclic RGD peptide. It's a ligand that's recognized by this integrin receptor that's overexpressed on many cancer cell lines. So the idea is if we decorate our nanoparticles with targeting agents that are specific for the disease tissue, then we can get improved uptake. And in fact, that's what we see, about a 10-fold increased uptake of this decorated object compared to the one that's not decorated. So the penultimate example I'd like to describe to you was done by Sean Douglas. You might have recognized his name from before. He was a graduate student who trained with me, and then he stayed on as a postdoctoral fellow to work with George Church to then apply the methods that he had developed in my group now towards this therapeutic type goal. And now he's assistant professor at UCSF. So the idea is that he used DNA nanotechnology to fabricate this nanoscale barrel, again with dimensions on the order of 50, 60 nanometers per salient dimension. And then he placed on the inside of this barrel a large number of therapeutic antibodies. And these antibodies had previously been known from the literature that if you present a large cluster of them to the cell surface of some lymphocytes from the blood, then that can basically trigger receptor clustering and then cause that immune cell to commit suicide. So this is a very important function within biology, that cells, when they're told to die, they die. And this can be used to combat infection, could be used to combat cancer. But you don't want to be presenting this cluster of antibodies to all cells and telling them all to die. You want selectively to only kill the diseased ones. And so in order to achieve that selectivity, what he did was he put these locks on the barrel. So in the barrel state, the antibodies cannot be reached by the cells. They're basically hidden from the cells. And this barrel has to be opened up for those antibodies to be exposed to the cells. And so what he did was he applied these molecular locks on the two ends of the barrel, each recognizing a distinct receptor that's overexpressed on the cancerous cell line, but not on the healthy cell line. And so what happens is if this molecular robot encounters a healthy cell, then it won't be able to unlock this lock or this one. It'll stay closed, and the healthy cell will be spared. But if the cancer cell encounters this, it will have both of the locks required to break these open. And this is a logical AND statement. So only if you have receptor 1 and receptor 2 will the robot open and then deliver the antibodies. So this could be important because maybe one lock is not specific enough. Maybe if we're only querying one surface receptor, it's impossible to avoid damage to the healthy tissues. So if we can make the decision-making process more stringent, then we can get better selectivity. And in this way, at least in a test tube, he found indeed that this logic-gated DNA nanorobot seemed to selectively kill the cancer cell line and not the healthy cell line. A few caveats. So this is only in a test tube. It's a much more difficult job to get this to work in vivo. There's all kinds of barriers. One of them is that when you build structures out of DNA, then it's susceptible to damage by enzymes in circulation that chew up DNA. So in order for us to actually use this clinically, then we need to find some way to protect them. And so another issue is maybe sometimes you want to have a target that's inside the cell. You don't want to just signal to the cell the outside. So here's a result from the labs of Hendrik Dietz, you might remember his name as well, and Fritz Simmel, where they programmed a three-dimensional DNA nanostructure to actually punch a hole into liposomes. So this kind of represents a model system for a cell. So this DNA nanostructure that they put these greasy cholesterol groups on actually will self-assemble and punch a hole into these vesicles. So you can imagine that this could be the genesis of some kind of therapeutic device that can punch holes into the disease cells and then very efficiently deliver drugs there. But again, with all of these toy examples, you run into the practical problem that they'll just get destroyed in serum. So the last thing I'd like to tell you about is a method that is addressing this problem developed by Steve Perel in the group. And so what Steve did was he was inspired by viruses that are enveloped by a lipid bilayer. So it looks kind of like a little cell. And the notion is that lipid bilayer is actually protecting the insides from damage by proteases and nucleases of the body. So could we try to use the same kind of strategy to protect our DNA nano devices from destruction? So in this example, what he did was he self-assembled a DNA wireframe octahedron that's about 50 nanometers in diameter. Again, he decorated it with these single-stranded extensions, in this case, all of the same sequence. Then he hybridized on a DNA strand of complementary sequence with this hydrophobic lipid group on it. So this is the building block of the lipid vesicles that surround our cells and envelope viruses. And he solubilized that with a detergent. So you end up with this object that has, in this case, 48 of these lipid molecules hanging off of it, and then it's kind of dissolved in a detergent. And then the next step was to actually get a liposome around that. So he mixed that with a solution of giant liposomes, just this bag of membranes. And then he dialyzed out the detergent, so kind of basically diluted out the detergent. And through some process that we don't quite understand, but we're excited about, you actually get shrink wrapping of the lipid membrane around our DNA nanostructure. So here are electron micrographs of the naked DNA octahedra. And here are electron micrographs of the encapsulated DNA octahedra. And these look almost identical to envelope viruses that you find from nature, but they're completely synthetic. They're not infective, and they can't do anything like that, but we think it's an important first step. So what Steve demonstrated is that when you encapsulate the particles, they're now resistant to degradation by nucleases, because those nucleases cannot penetrate the lipid bilayer. They're also, they don't seem to be taken up very well by immune cells. So it turns out one of the other problems with DNA as a therapeutic is that our immune system is trained to attack and damage DNA, but when you wrap the lipid around it, you protect these from engulfment by macrophages and splenocytes, and they don't really seem to set off that immune activation. And finally, Steve also injected these into mice to see what would happen to them. Again, the problem that we're trying to avoid is destruction of the DNA in circulation. So on the far left is a negative control, where we take a fluorescently labeled DNA oligonucleotide, so just short strand of DNA, and then inject it into the tail vein of the mouse and look at its biodistribution after 2 hours. And what you can see is that, or we can infer, is that DNA gets degraded, passes through the kidneys, and just accumulates in the bladder. So it's a fluorescent label. And then in the middle panel, we take our DNA nanostructure, and we label that with the fluor. And after 2 hours, we see the same fate, that it's getting degraded, goes to the kidneys, accumulates in the bladder. But now with the encapsulated DNA structures, after 2 hours, we see very nice biodistribution of the object. And we're quite excited about this because maybe it means we actually can use these DNA nanostructures towards therapeutic type ends. It's still early days. And so in conclusion, hopefully I've persuaded you that we have this amazing new capability to structure matter on this very small scale. What's remarkable is it's basically like digital control over the basic stable building blocks of nature, atoms and molecules. And we can control exactly where each atom is going. Hopefully I've given you a sense of why DNA itself is so well suited for this process, that you have this robust interaction, A pairs with T, C pairs with G, that you don't see with any other known material thus far. And hopefully I've also persuaded you to think about how this kind of control actually could be useful towards practical ends. So I'd like to thank the following sources for support, many of them with offices in this area. So I'm often coming down talking with program officers. The group of investigators in Brown here, former graduate students and postdocs who really developed the underlying technology, and now most of them have their own labs around the world. And I think I'll just stop there. Thank you very much for your attention. I'd be happy to take any questions that you have.
Thank you very much, William. We have 2 microphones going around. Please raise your hand. Wait for the microphone. State your name. You stand up, state your name, and mention whether you're a member of the society or not. Yes. Yeah.
Bob Hershey, I'm a member. What is going to be the next step to get these for forms that they can be useful to us?
Yes. So I would say with any new technology, there's lower hanging fruit, and then there's things that are further away. So I'd say the things that are closest to us are probably removed from immediate concern of a layperson. So for example, you could ask the question, what's the point of molecular biology to further tease apart the mechanisms of cells? Because realistically, most of the discoveries that we make today are not going to be translated to the clinic in the next 10 years. So the answer is really that it's really preparing for clinical results 30 years from now. So if we look at the amazing advances in molecular medicine today, that we have this specter of personalized medicine. There's a drug called Gleevec, maybe you guys have heard of. It's basically a miracle drug for people who have a particular kind of leukemia. So before this drug existed, it was basically a death sentence. And when they were developing the drug, in fact, Novartis was initially thinking, well, there aren't that many people suffering from this kind of cancer, so maybe it's not profitable for us to pursue it. But they decided to pursue it anyways. And now it's a big blockbuster drug for the company. And do you know why? It's because it's been so successful in keeping people alive. Now there are a lot of people with the disease, and so there's a big market. So I think basic science is incredibly important for the long-term health of technology and people. In terms of, you might ask, well, what's the nearest term advance that we could get that would be directly relevant to human health? So I think there could be some things related to diagnostics. So maybe if, I'm just speculating here, but you could imagine how early detection of cancer is an extremely important problem. And so there's a lot of interest in coming up with methods for more sensitive detection of the markers for disease. So you can imagine how, I'm not going to go through the details, but how this kind of nanoscale control might be exploited for more precise biodetection of some of these markers. So that's something you can imagine would happen sooner. It also potentially could avoid all the difficult regulatory issues. So if you're trying to introduce a drug into the body, you have to pass all kinds of hurdles from the FDA in order to actually pursue study. But if you have a diagnostic that you're using on a drop of blood that you're taking from the patient, then that's something that you can get into actual commercial use much faster. In terms of a therapeutic, so I mentioned this lipid encapsulated structure is something that could basically circulate around the body and hunt down pathogens and that sort of thing. But to be honest with you, I don't think anything like that is going to be available in the clinic for probably at least a few decades, because there's all kinds of challenges there. Our body is very efficient at removing foreign particles from our bloodstream. It's just a really difficult problem. We are, in my lab and at the Wyss Institute, we are working on another project that technologically we think is more attractive in the near scale, which is that we're trying to vaccinate against cancer and other diseases. In other words, we're trying to stimulate the most advanced nanotechnology known in the universe, which is our immune system, which currently is probably centuries ahead of what we can do in the laboratory. And we just want to help out our immune system. So the idea for this vaccination, it's actually a local vaccination idea, is that we're going to create an artificial wound site in a local part of the body. So let's say just a subcutaneous injection of some basically tumor lysate. So the surgeon will cut the tumor out, grind it up, and within that tumor lysate, it'll be an overabundance of some of the pathogenic proteins that tumor is using to be successful. And then you embed that into a part of your skin close to a lymph node, and you formulate in a way using this nanotechnology to make it extremely aggressive in terms of stimulating our immune system to hunt for that and kill it in our body. So the way our immune system works is it's kind of like there's these killer T cells, they're like trained hunting dogs. And so what you do at a lymph node is you present it with the smell, the thing that it's supposed to kill. And then once it gets a whiff of that, then it becomes activated, and then it'll expand, and it'll basically search, hunt, and destroy. And it's extremely efficient at this. So efficient that every one of us in this room has tumors in our body that our immune system is cleaning up, and so we don't notice because it's doing its job. But it's just in the case where the tumor is able to set up a local immunosuppressive environment that in a few cases it's able to overcome the natural function of our immune system. That's when we notice we have a disease. So can we fight back by giving our immune system a little bit of an extra boost to overcome that? So I believe that in the next 10 to 20 years, nanotechnology might actually play a role. We could actually see something in clinical trials in the next 10 years or so that maybe could help by basically piggybacking on a more powerful technology than we currently have. But then looking longer scale, something like 50 years, 50, 100 years from now, at our current rate of progress, I'd like to believe we'll actually be building things like the immune cells themselves. So we could have a completely artificial robotic nanodestruction system for directly hunting down disease. So that's my hope.
Well, okay, go ahead here. Oh,
Just for giggles.
could you state your name, please, if you remember? State your name and if you remember or not.
My name is Walter Lee.
Thank
and
you.
I'm a private citizen who came just because it sounds like interesting stuff, and it certainly was. When you find one of these things and you tag it as a thing to be removed, can you hear me now?
Yes, thank you. Thank you.
I've forgotten the question by now. When you tag these things and send something in to kill and pick out certain cells it wants to get rid of, how does it pick out those cells and forget everything else?
Yes, so basically our cells, all of our cells in our body have evolved to cooperate in order to help our immune system figure that out. So you could say, what if there's a pathogen hiding inside of a cell? How can our immune cells actually detect whether or not that pathogen is inside that cell? And so what the cells in our body have evolved is a mechanism to help out our immune system. So they have these machines called proteasomes, they're like molecular wood chippers. And they just basically, they're randomly cutting up any protein that it encounters inside of the cell, and then it's exporting it to the surface of the cell. So basically, every single one of the cells in our body is displaying on its surface a bunch of little molecular wood chips that represent the destruction of everything that was inside of the cell. So that's just something we've evolved. And now what happens is you have these immune cells come by, and then they start sniffing the outside of the cell. And they can say, okay, do I smell something that looks like what I recognize? And if it recognizes it, then it starts to get excited, starts to expand, and it starts to kill cells like that. And so you might say, well, that doesn't make any sense. If you have these wood chippers that are chipping up everything in the cell, and it's displaying it on the outside of the cell, and you have these immune cells will attack things they'll recognize, won't we get immune systems that will just attack everything? And in fact, we do get that. That's autoimmune disease. So that's a huge health problem. Probably most of the diseases of the modern world have something to do with autoimmunity. So it's a real thing. But our body tries to fight that during development within, for example, within the spleen and the blood marrow, and the bone marrow. Our immune system does this amazing thing where it grows up all of these immune cells, each one that recognizes a different little fragment, and it decides to kill all the ones that recognize the self-antigen. So it's called self-selection. So it tells any immune cell that recognizes a self-antigen to commit suicide or basically don't attack that. And so you're left over with immune cells that will only attack foreign particles. And so how does that, so certainly that makes sense in terms of a viral infection. So a virus comes into a cell, it gets put through the molecular wood chipper, parts of the virus get displayed on the outside of the cell. Now you're going to have lymphocytes circulating that will recognize the thing that isn't present in our normal repertoire, and then you get specificity. It's more challenging when you think about cancer because at the end of the day, a cancer cell is one of our cells. And so all it's displaying are our own proteins. So it's a little bit more complicated, but you could imagine that the cancer cell might be displaying a vastly increased number of those antigens. So our immune systems have evolved to only get excited when it sees lots of those things. So for example, one common mutation is overexpression of epidermal growth factor receptor. It's this receptor that basically signals the cell to start dividing, which is what cancer cells like to do. And so many cancer cells overexpress this receptor. So they have a huge abundance of this receptor on the cell surface, and also pieces of it that are displayed on the cell surface. So maybe the immune cell, if it just sees a cell with a couple copies of this, then it won't get excited. But if it sees many, many copies of it, then it will get excited. So that gives you a little bit of the flavor how we think it's able to achieve specificity. But it's important to realize it's not infinitely good at it. So we do have immune evasion on one hand, where it's not basically not able to recognize that, and on the other end, we have autoimmune. So our immune system's kind of in between a rock and a hard place. If it's not active enough, then we're going to suffer from infection and cancer. If it's overactive, we're going to suffer from autoimmune disease. And where I think medicine of the next century will come in is it will help the immune system to locally be more aggressive towards specific threats. Right? So currently, immunotherapies of today, they're mildly effective, but they're kind of like blunt weapons. So you either globally upregulate your immune system or globally suppress your immune system. And it's just not, it's not the most precise strategy. It's just the best we have today.
Another question? The lady there.
Hi, I'm Erica Olander. I'm not a member here. I have a question about, I'm sure someone in your lab has been thinking about whether these structures are biologically active in the cell, and I know that they're barely getting into the cell, let alone the nucleus. But in vitro, would an RNA polymerase hook up with these structures and start to transcribe them?
Yeah, I think that could be very, I mean, that question is fascinating on two levels. So one level is, can it cause trouble? Can it go in, integrate into the genome? That can cause cancer. And then the other side of the question, which is also maybe even more interesting, is can you actually exploit that? So can you get some structure that will go into the cell and do specific gene therapy or do specific knockout of transcripts, like with siRNAs, something like that. So I think the second aspect is to me, the most interesting aspect, because that really implies a huge level of control over the behavior of the cells. So I believe that will be a major direction, actually, of controlling transcription of things inside of cells to modulate their behavior. In terms of the other side, which is potential side effects, I think with any material, any material has a potential damage. If you drink too much water, you can kill yourself, right? Oxygen, if you have too much oxygen, you can kill yourself. I think the important thing is to recognize where on the spectrum of danger your material lies and what you can do about it. So we would say, well, water, we kind of need it to survive, and it's actually, on the scheme of things, it's not that dangerous, so let's not tell people not to drink water. But on the other hand, you have things like Ebola virus, and you can say, well, that's pretty bad stuff. We better keep that away from people. Or maybe you have some asbestos or metallic nanoparticles or carbon nanotubes, things that they might be good, but you're kind of worried they'll accumulate in the body and they'll do bad things. So I would say DNA nanostructures are somewhere kind of more in the middle, more towards the benign. They are biocompatible. They will degrade, so that's an important thing. A lot of therapeutics, you want your therapeutic to eventually go away. You don't want it to stick around forever. So that's the nice thing about DNA, is it won't stick around forever. In terms of its ability to integrate into the genome, I think the evidence thus far suggests that's not a very easy thing to do, because most of the time people are actually trying to get things to integrate into the genome. But you should worry about it. And what I have confidence is that the extreme programmability of DNA will allow us to engineer around those kind of threats. So I think that the benefits for a material like DNA far outweigh the potential problems. So sure, there will be side effects of this, like any medicine, but I think it's much more on the benign. I think the benefits of DNA far outweigh the potential harm, because it is a biocompatible material in the end.
Another question? Let's see one in the back there, the gentleman.
Hello, Dr. Shih. Joel Wilson, just a private citizen. I was wondering about the possibility of this technology being applied to other basic or similar basic questions in science. For example, one of the primary ones in protein chemistry is, why do proteins fold the way they do? It seems like this would be ideal. Have any other investigators looked at this to try?
Yeah, I think that's really, thank you for the question. I think that's a really fascinating question and a great direction for the field that people are looking. So you can imagine these structures addressing that question from many different angles that I think are very interesting. So one is as a model system for a polymer folding into a structure. So they're not proteins, but DNA structures they have much more in common with protein than they do with iron or carbon nanotubes. They're both kind of floppy polymers, about the same Young's modulus as plastic. They're using weak interactions. They're soluble in water. So you can imagine, because our structures are much more easy to analyze than proteins, we could use them as a model system for general biopolymer assembly. So that's one area. A second area is that just protein structure. So in order to understand how a protein folds, it's helpful to know what it's folding towards. So I mentioned one example of how to solve the structure of proteins with our DNA tool. There's also the second method that Ned Siemens been trying to do with host-guest crystallization. I think there's going to be a revolution in methods of determining protein structure, and maybe also following protein folding, that, that this kind of precise nanotechnology will be able to avail us to. Another idea is that it can be hard to study protein folding because the intermediates of protein folding tend to be have their greasy part sticking on the outside. They tend to aggregate, and then you can't study it. But we know that within cells, cells can get around this problem by, in some cases, folding the proteins within these little nanoscale compartments called chaperones. So maybe we could build DNA-shaped chaperones or cages to sequester, maybe that's a bad word in this town, to put proteins on the inside and prevent them from aggregating so we can better study them. So I think that's a really fantastic area for the field. Thank you.
Lloyd, go ahead.
Question. Lloyd Mitchell. I have a question about, has anyone been using these to, like, say, combine the property of nanotubes to change the tropism of viruses, which have evolved very well to, inject genes and things like that, but you could use this to perhaps change the tropism to different bacteria or cell types?
Yeah, so I kind of presented this as DNA or bust. So DNA is really the main player, the superstar of the therapeutic toolkit, but really we should be combining the strengths of many different materials. So I don't know if this is exactly what you're asking, but for example, could we re-engineer viruses for gene therapy, or maybe want to engineer viruses to attack cancer cells, something like that. So for example, maybe you could use the DNA nanostructure to make a kind of hybrid device so that the virus has its own ligands for binding certain targets, but then you graft on something else that increases or somehow modulates its binding affinity for the target. You could imagine another problem is that if you use a virus as your carrier, then you probably want to change its cargo. So it normally has its genome in there, but for therapeutic purposes, you don't want to inject its genome. You want to load it up with drugs. So maybe you could imagine, and this is not a trivial problem, but you could imagine some molecular scheme where you basically cause the virus to eject its contents, and then you construct a nanoscale pump, maybe out of DNA nanostructures, that uses some energy source to actively pump the drug into the inside of the virus. So both change basically the cargo, and then maybe also the selectivity of a virus to use it as a therapeutic tool or research tool. Is that addressing your question?
Okay, let's say the lady in the back there.
I'm Janet Moss and a member. Have you looked into superoxide dismutase and the NRF2 activator in the cell and how it eventually affects the DNA?
We have not looked at that. I'm thinking, I remember vaguely as a postdoctoral fellow, there was some idea of using what are called DNA zymes in order to, I believe, to knock down superoxide dismutase.
5 enzymes.
And I don't remember the details, but this is just an example to mention that everything I told you about is basically double-stranded DNA, which is kind of inert in terms of specifically binding to different proteins if you want to inactivate them. But it turns out DNA has another capability. You can evolve in the test tube a single strand of DNA. If you leave out the complement, then you can evolve sequences that will fold by themselves into complex three-dimensional shapes. And then just like proteins, you can evolve them to have selective binding or catalysis onto protein targets. In this case, there is a single strand of DNA that was evolved in the laboratory to specifically cut RNA strands in a sequence-selective fashion. So what people have been thinking about is, can we use these DNA zymes to cut the transcripts that drive expression of these enzymes, and then maybe have some kind of benefit on that level? But that's as good as I can give you for.
Yeah, there's
that.
also research on telomeres. Have you on the ends of the DNA?
Oh. I haven't really thought about it. You could imagine, like maybe your goal is to extend the length of telomeres in cells to basically decrease their senescence, or vice versa. You want to chop off their telomeres to help them die. So you can imagine nanoscale structures that, by virtue of their shape or some kind of active motility, can specifically target to go into the nucleus. So that's not so easy to get things to go specifically into the nucleus, and then maybe through some kind of locomotion or just molecular recognition, can target the ends of chromosomes or the middle of chromosomes or wherever it is that you want, and then mediate some kind of effect there. So I think that's an exciting idea. I'm not yet aware of anybody trying to do that. It's also a difficult thing to get things to go all the way into the nucleus, but it's a fascinating idea, though.
Let's try a couple more questions. Let's say the gentleman in the back and then you. Okay.
Hi, I'm Alex Shaitan. I'm not a member. I have a question concerning the physics of DNA nanostructures formation. Actually, DNA is heavily negatively charged, right? And the negative charges, they should repel. We know that in the nucleus there are specific proteins, the histones, that are positively charged. So how can you comment on this problem?
So in fact, that's absolutely correct. And we need to have a counter-ion to stabilize that negative charge. And our most effective cation is divalent cations, actually. So we like to use magnesium. It's present in cells. Actually, the amount of magnesium that's in cells isn't quite enough to stabilize those structures. So we're looking for other things that could substitute for in vivo functions. But in a test tube, we just know if we add enough divalent magnesium, then we can get folding. So if we add too little, then the structure, there's too much repulsion, it never folds. It turns out it's a problem if we add too much, because then now we start to favor aggregation. So aggregation now becomes more favorable than our proper structure formation, because to magnesium ion, an aggregate looks about the same as one of our DNA structures. So we have to tune the magnesium concentration exactly right to get the desired folding. We've seen some interesting behavior, such as that if you try to add monovalent cations and divalent cations together, then you actually slow down the folding of the structures the more monovalent cations that you add, which at first seems kind of counterintuitive. You should think, well, if I had more monovalent, shouldn't that do a better job of neutralizing the charge? And so what we think is happening, people have actually seen this in polyelectrolyte condensation before. And so the idea is that the monovalent cations actually outcompete the divalent ones. So they prevent the divalent ones from coming in and getting the folding to happen. And then you can get another interesting reversal. Once you go up to really high monovalent concentrations, then it can start to promote folding again. So low monovalent concentrations suppresses folding, but really high monovalent concentrations promotes folding.
And one last question. You have a question, Mr. Yeah, go ahead, Mr. Rosen.
You're talking a lot about doing these things, using these things against cancer. Oh, David Rosen, lifetime member. How about things like pathogens and infectious diseases, or even pesticides, something specific to get rid of specific insects or mice or something? Have you thought about applications like those?
You mean both inside the body and outside the body, or just?
Yes, outside the body. Yes, both inside and outside the body.
So first.
You keep on talking about cancer.
Right. Yeah. So maybe cancer's not the biggest problem, but it's the one with the most funding, and it's one that we all care about, even if it's not the number one killer. So I would say for pathogens, it's kind of very similar. So like with cancer, I mentioned two different ways you could attack cancer. So you could either just give your immune system a boost and let the immune system do the heavy work, or you could imagine directly attacking the tumor. So you could have a nanoscale particle that circulates in the blood that targets, that goes into the tumor and tries to kill it, or you could try those things in combination. And the same thing is true for pathogens as well. So you could imagine creating nanostructures either that prime the immune system to recognize and attack those pathogens, because a lot of pathogens that likewise have evolved immune avoidance. They have all kinds of strategies for escaping our immune system. So it's an arms race. So we can imagine medicines that help reboost our immune system against those pathogens, or we could have things that directly target the pathogens if you can figure out a way to do that. Outside the body, pesticide. I mean, I haven't really thought about that. I mean, one issue with something like a pesticide is presumably you need a lot of it. And currently, DNA is not so cheap for us to make. So like the cheapest we can make on a synthetic scale is something like $1 million per kilogram, which is cheap enough for some therapeutics. But you can imagine maybe for like a pesticide you're trying to spread across a large area, that's not going to be economically possible. I think eventually, decades from now, we'll use genetic engineering to program bacteria or algae or something to or plants to basically extrude out DNA at extremely low cost. So instead of making cotton, they'll make DNA. Because from a fundamental point of view, there's nothing in DNA that's that expensive. Carbon, nitrogen, oxygen, phosphorus is a little bit expensive. But basically, DNA is not that expensive in terms of its material constituents. So it's conceivable we could program, use synthetic biology to basically lower the cost of DNA down to the cost of cotton. So if you have a pesticide that's the same cost as cotton, then maybe that would be cost effective. But yeah, I haven't really thought about it in that much detail.
Well, thank you very much. Before you leave, we have one more thing for you. It's a coveted PSW plaque. It's the announcement of signed and framed by all the members, signed by all the members of the General Committee and framed for you to keep. Again, thank you very much.
Thank you.